dengue, Zika, yellow fever, chikungunya. For diseases, one shared vector, the 𝗂𝐸𝑑𝑠 𝗂𝗎𝑃𝑡𝑒 mosquito. This is the story of an engineered answer to an old problem. 𝗂𝐸𝑑𝑠 𝗂𝗎𝑃𝑡𝑒 thrives close to humans, biting by day and breeding in bottle caps and tires. Its eggs survive drought for over a year. Since 2013 it has spread across new U.S. regions, resisting sprays and larvicides. One old idea, sterile insect technique. Irradiate males, release them to mate uselessly. It eliminated the screwworm fly, but irradiation weakens mosquitoes. Sterile males compete poorly for mates, blunting the technique in the field. ℧𝗏𝑐𝑠 answer, RIDL, release of insects carrying a dominant lethal gene. In the lab, tetracycline keeps that gene switched off, so mosquitoes develop normally. Released into the wild, without tetracycline, their offspring inherit an active lethal gene and die before adulthood. Only males are released. Males don't bite, they feed on nectar, not blood. Sex sorting engineered mosquitoes before release, so no biting females reach the wild, has been a central engineering challenge. Generation after generation, sustained releases compound. Each cohort of wild offspring inherits the lethal gene and fails to reach adulthood, a numbers game that, sustained over time, drives the local population sharply downward. Grand Cayman hosted the first open trial in November 2009. Brazil's field trial, reported a success in January 2016, built confidence in the technology just weeks before Zika fears put U.S. mosquito control back in the spotlight. Science demands honesty about limitations. A 2019 study found genetic material from released 𝑂𝑥513𝐴 mosquitoes had persisted into Brazil's wild population. Headlines called the mosquitoes more robust. Scientists pushed back, the real finding was gene persistence, not tougher or more dangerous mosquitoes. ℧𝗏𝑐𝑠's next generation, 𝗂𝑥513𝗏, refines the approach with a female-specific lethal gene. Males survive and pass the trait on without a tetracycline rearing cycle for every release. The EPA granted an experimental permit in June 2020, with Florida trials approved for 2021. Compared to broad-spectrum spraying, the case is straightforward, species-specific, so pollinators and other insects go untouched. No chemical exposure. Scalable. And targeted precisely at the one species actually spreading disease. This has been an independent science explainer, not an official ℧𝗏𝑐𝑠 production, grounded in 𝗧𝗏𝗏, Nehera, James, 𝗩𝗏𝗏, and Bloss, 2021, Pathogens and Global Health, full citation and DOI on screen, with ℂ𝗏𝑐𝗏𝗏 details drawn from the wider public record.